Hi, everyone, and thank you for joining me today. I will be presenting for the MassURC, the research that I've been conducting at UMass Amherst under the Rao Lab for the past year and a half, as well as the research that I'll be writing my undergraduate thesis on. So today I will be talking about remote control of neural activity via magnetic stimulation. So just a little bit of background. There are two main strategies used for the control of neurons: magnetomechanical stimulation and magnetothermal stimulation. And as their name suggests, magnetomechanical stimulation uses magnetic nanoparticles to apply a small force on cell membranes. And magnetothermal uses magnetic nanoparticles to apply heat to the membranes. The ultimate goal of both strategies is to depolarize the neurons by activating calcium ion channels to open and allow for an influx of positive ions into the cell. Magnetic nanoparticles are the vectors that relay the stimulus, and the stimulus is delivered via an alternating magnetic field. Now, my cell cultures have consisted of human embryonic kidney cells because they are more resilient than neurons and easier to transfect, and they act similar to neurons when they go through the transfection process. Now, there's a two-stage transfection process needed to prepare these cells for the magnetic stimulation. The first stage transfects a calcium ion channel sensitive to the stimulus delivered by the magnetic nanoparticles. Now, such a calcium ion channel would be TRP1, for example, that is thermally sensitive, or TRP4 and piezo2, which are both mechanically sensitive. The gene maps for this plasmid for TRP4 and TRP1 are seen in the bottom figure. This transfection uses a fluorescent protein marker, M-cherry, to indicate a successful transfection, and this can be seen in the top right image. You'll notice that it glows a fluorescent red color. The second transfection uses GCAMP, a calcium indicator. So while the first transfection makes the stimulation possible, the second one is so we can visualize the stimulation happening in real time. This transfection will allow the calcium in the cell to light up a fluorescent green color, which is indicated in the middle image. So as discussed on my previous slide, GCAMP is a calcium indicator. So ideally we would get data similar to the graph to my left. The images on the bottom show cell cultures after both transfections and after being treated with magnetic nanoparticles. The image with less fluorescence on the left is before exposure to an alternating magnetic field, and the image to the right is after exposure. This change in fluorescence is indicative of successful remote-controlled cell depolarization. This data can then be quantified as change in fluorescence over time, graph or chart, using programs such as MATLAB. The ion channels that are being transfected depend on the stimulation strategy being used. TRP1 is used for magnetothermal stimulation, and TRP4 is used for magnetomechanical stimulation. The protein channel piezo2 has also been known to be used for magnetomechanical stimulation in other trials. The physical properties of the magnetic nanoparticles that the cells are being treated with, along with the properties of the alternating magnetic field that the cells are exposed to, determine whether the nanoparticles will produce a mechanical force or heat. Magnetic nanoparticles around 200 nanometers in diameter with a slightly more disc shape are ideal for mechanical stimulation. Magnetic nanoparticles that are around 20 nanometers and spherical are more optimal for thermal stimulation. So here's my current magnetomechanical setup for producing low-frequency alternating magnetic fields. The setup includes a waveform generator, which produces the initial sine wave signal, an amplifier, which strengthens the signal, a hollow copper solenoid coil to run the current through and produce a magnetic field, and finally an oscilloscope to measure the output from the coil and the amplifier. The coil is placed on our inverted fluorescent microscope stage, and the cell cultures are placed within the coil. This allows us to record microscope footage while the cells are actively exposed to an alternating magnetic field. One problem that I had to overcome was that the strongest point of the magnetic field is actually in the middle of the solenoid. So I needed a way to raise the cell culture to closer to half of the coil's height. While this could be done with a smaller stage inside of the coil with a clear bottom, I also wanted to make the height of the cell culture adjustable, since focusing the inverted microscope's objectives would be difficult due to the coil's bulky size. I overcame this problem by using SolidWorks to design a holder for a cell culture dish. The holder was 3D printed and mounted on the coil, and thanks to this holder, the cell culture dish can now be adjusted to any height within the solenoid. Here we have a field map of the magnetomechanical solenoid coil shown in previous slides. Using the program Finite Element Method Magnetics, I was able to simulate a scale model of the coil with identical material properties and input currents. The resulting field map shows the values that are consistent with the readings on our Gauss meter. Running at about 2 amps of alternating current, the coil can put out our target field strength of 23 milliteslas. Through testing the upper limit of the amplifier's output, we've been able to see the coil put out over 75 milliteslas. Since the field map is to scale, we can use it in conjunction with the Gauss meter to measure where the field is 23 milliteslas in terms of the coil's height. And we can coordinate that with the cell culture holder height. The magnetothermal coil involves a much more complex design. This is due to the extra power requirements. This coil setup uses a ferromagnetic core toroid instead of a solenoid. The resulting magnetic field is stronger, but only covers a very small area. There are also drawbacks due to heat dissipation. A cooling system, usually involving cold water being circulated around the coil, is required. This strategy needs a much higher frequency, in the hundreds of kilohertz, of magnetic field strength to heat up the magnetic nanoparticles. The higher frequency makes the field require much more power. The setup requires a more powerful amplifier, a step-down transformer, and a high-voltage capacitor array. Our setup is still being built, but these images are what a completed setup would look like. With the completed magnetothermal coil setup and successful magnetomechanical trials in view, we can start to look at future applications of this technology. In vivo trials for the magnetomechanical setup would be the next step, since that coil is already large enough to fit a mouse or rat. This would allow for behavioral studies and live tissue cultures to see how magnetic nanoparticles affect brain physiology. And for the magnetothermal in vivo setup, this would require an even more complicated setup than the corresponding in vitro coil. To scale up the size of the magnetic field area, a full bridge inverter would be needed to boost the voltage using DC power and then converting it back to AC. Thank you for watching. I hope you come and see my poster session at the MassURC on April 22nd.